Hello, everyone. I'm Jun Kim, a TA for the Intro Bio class, and in this video, I want to help you guys figure out how you can learn and approach the learner's manual questions. So, there are several common concerns for a lot of Intro Bio students, specifically regarding the questions in the learner's manual. For example, I've heard a lot of people be frustrated about the fact that they feel that the exam questions, for example, and the learner's manual questions are very different. So they solve the learner's manual problems, try to learn from them, but when it comes to exam time, um, the unfamiliarity of the exam questions kind of throws them off, and it gets them frustrated. And a second common concern, for example, is the fact that there's so much to take away from each of the learner's manual problems, especially from the solutions. As some of you may know, the solutions to some of the learner's manual questions are actually longer than the problems themselves in terms of how much text and information there is. So that can be, that can throw some students off and add to the confusion. So in this video, I'm trying to offer one possible solution, and by no means this isn't the only way to approach you know intro bio at Columbia. But this was what worked for me, and it worked for a lot of other TAs that I've talked to. And there's two points of emphasis here. The first thing is that you want to take away the general principles, the larger concepts of biology from the problems, instead of like all the specific nitty gritty details that are really applicable to only one problem, right? And by doing that, what you can do is, instead of learning the specific case by case scenarios, if you learn the general laws and the principles and concepts, and you can take those and apply them to exam problems, which again are not exactly the same as the learner's manual problems. And the second thing is to have a running record, log, journal, some form of documentation, where you jot down the principles that you kind of missed the first time around while solving the learner's manual problems, and that would that would be your review sheet before exam time, instead of having to resolve everything, for example, or re-memorize everything in the lecture notes. And the way I'll be doing this is through two sample problems. I'll solve 7R10 and 9R7 as if I'm an intro bio student approaching that for the first time. So with meaning some degree of familiarity with the content of the behind these problems, but once in a while making a few logical missteps, making some errors here and there, and then more importantly, what I'll do is show what I'll be writing down in my learner's log or learner's or lesson log, and so that even though I'm only presenting two problems, ideally what I'm hoping that you guys can take away is how you yourself, when solving the learner's manual problems, can extract the principles that the problems want you to take away. Okay, so let's begin with the first problem, 7R10. And by the way, this is in the hormone unit. So how I approach each of these problems is by reading the STEM or the prompt very in depth the first time around. And I think that's the best way for me, because there's a lot of information that's in the problem that I need to utilize to solve the rest of the answers. Okay, so GH, or growth hormone, has long-term effects on growth that requires the synthesis of IGF, or insulin-like growth factor. Part A, IGF stimulates the growth of many cells. It uses a tyrosine kinase receptor and stimulates the MAPK pathway. When IGF binds to its receptor, the receptor itself, without any other proteins, should be able to do which of the following? There's a lot to take in here, but let's begin by first drawing out what we know, or what we can take away from this Part A problem stem. So the question mentions a tyrosine kinase receptor, right? And I'm going to go ahead and draw the cellular membrane right here, right? And I drew two lines because each of these represents one leaflet of the plasma membrane of the cell, right? Now remember, the plasma membrane has two leaflets, the extracellular one and the intracellular one. So although I drew 2 lines, really I'm only drawing one membrane. So here's a plasma membrane, and the question mentions that the IGF receptor is a tyrosine kinase receptor. So I'm going to draw the tyrosine kinase receptor like this, with the extracellular domain that binds IGF and the intracellular domain that engages in the signaling downstream of whatever happens beyond after IGF binds. And the question does mention that beyond this point we have the MAPK pathway. So MAPK pathway. Okay. Now let's recall what happens with tyrosine kinase binding and binding its ligand or IGF and its activation. So I'm going to draw IGF in red, coming in like this, right? And when that happens, right, we can go back to our class notes about tyrosine kinase receptors, and we then remember, you actually need 2 tyrosine kinase receptors to bind their ligands, each respectively, like that. And afterwards, what you get is a dimerization, or if I were to draw it here, you would get 2 TRKs with their ligands bound, interacting with each other, right? Something like that, right? And again, each of them bound to their IGF molecule, like that. Okay, so let's try to remember what specifically happens after tyrosine kinase receptors dimerize and activate each other. Well, specifically what happens is, the activation involves phosphorylation, or the addition of phosphate groups to the intracellular domains of each of these TRKs. They phosphorylate each other. So I'm going to draw it like this in blue. P in the circle, those are all the phosphate groups that gets added. And notice, again, these are all on the intracellular domain, because that's where all the intracellular signaling is going to occur. And once you get the phosphorylation, then these TRKs can then phosphorylate other proteins and then eventually you'll get activation of the MAPK pathway, right? And because of its ability to phosphorylate itself and other proteins, that's why they're called tyrosine kinase receptor. Kinase for defining an enzyme that's able to transfer phosphate groups, specifically adding phosphate groups to its substrate. Okay, so let's go back to the question. The question asks, the TRKase receptor without any other protein should be able to do which of the following? All right, part A, or answer choice A. Catalyze a trimeric G protein. No. G proteins are involved with G protein receptors, or GPCRs, rather than TRKs. TRKs, as we drew here, they don't involve G proteins. So I would rule that out if I were to solve this problem. Second, or B, dimerize. Yep, that sounds about right. I mean, that's exactly what I drew, and based on the lecture notes, that also seems to hold true. So I would indicate, I would write down B, or circle B. Let's keep going. Bind to DNA. Well, so the question asks specifically without any other proteins in parentheses. Now that may seem like an insignificant detail, but it's crucial here because although the MAPK pathway eventually leads to transcription factors binding to DNA and activating their expression, the question specifically wants, what do the TRKs themselves do? And they definitely don't bind the DNA at all. They're stuck in the membrane. So I would rule that out. D, phosphorylate its extracellular domain. No, we specifically mentioned that the phosphates, and also indicated in this diagram, gets added in the intracellular domain for the intracellular signaling. So I wouldn't circle that. Activate RAS. Now, RAS, if we remember from our class notes, or we can look it up also, is a small G protein. It's a little bit different from the other G proteins, but it's still categorized as a G protein nonetheless. And as we said earlier, there are no involvement of G proteins in TKR receptors. So I would rule that out. Catalyze hydrolysis of ATP. Well, I'm looking at this and I see phosphate groups transferred, but I don't really see ATP in this diagram. And, there doesn't seem to be any energy usage because that's what ATP seems to be used for, right? So, I'm a little bit hesitant to actually choose this as an answer. And since I've already found a good answer in B, I'm going to go with that's not F being not true. Essentially, out of all the options from A to F, I would only choose B. Let's see how I did. Well, it turns out I got half the answer, right? B, dimerized, we clearly knew that was going to happen based on the diagram we drew, and that was indeed correct. But F, the one we were uncertain about, seems also to be true, and we opted not to choose it, but that ended up being not quite true. So catalyzed hydrolysis of ATP. Let's think about this. Well, now why does that make sense, or why does that need to happen with the TKRs? And if we think about it, so we get phosphate groups that are added both to the TKRs themselves and to the proteins that get phosphorylated by the TKRs. And then we wonder, so where do these phosphates come from? And then we realize, a kinase doesn't just spontaneously generate a phosphate group. It has to get that phosphate group from somewhere. And that somewhere happens to be, in almost all cases for kinases, ATP, right? So a phosphorylation reaction necessarily involves the removal of a phosphate from an ATP, or AKA its hydrolysis, right? Okay, so what do we want to take away from this problem, right? So because we got part of the answer, but not all of it. And this is what I would write down. Well, actually, let's start with what we shouldn't be writing down. And that is, I have it written on the left-hand side of the PowerPoint in red, which is tyrosine kinase receptors not only dimerize upon ligand binding, but also hydrolyze ATP. Now, none of this is wrong. In fact, everything is correct. And in fact, this encompasses both of the answer choices, including the one we got, dimerize, and the one we didn't quite catch, which is catalyzed hydrolysis of ATP. But notice that the statement here on the left-hand side in red, it can only be applied to this specific problem about TKRs asking about ATP hydrolysis, right? And there's no guarantee that, for example, on the exam, you're going to get this exact question. In fact, you most definitely will not. So to make this exercise more useful, you want to take away the general principles, as I mentioned earlier in the introduction section of this video. So I have what I would write down as the general takeaway for me on the right-hand side in green and blue. Green meaning the content stuff, the content of the concept, or like the information that I want to take away, and the blue meaning indicating the kind of like the action, or like the action reminder of what I should do when approached by a similar problem on like later in the learner's manual or on the exam. So let's start with the green. Nearly all phosphorylation reactions in the cell require ATP hydrolysis for energy and phosphate. And then, for example, TKRs or TRKs. And this, notice, is a lot more general than the one on the left-hand side. It's discussing most, or basically almost all phosphorylation reactions, right? Not just the ones done by TKRs. And then it mentions that also you need the ATP hydrolysis specifically for the energy and the phosphate that you get from breaking off that phosphate from the ATP molecule, right? So this could be applied to a question that doesn't involve TKRs, but still involves a phosphorylation reaction, like basically any other kinase in the cell. The TKRs aren't the only kinases we have in the cell, but they all have to undergo ATP hydrolysis for the kinase activity to happen. And on the blue, we have make sure to check for ATP hydrolysis whenever phosphorylation is mentioned. Again, kind of like a reminder that we set for ourselves. Okay, so let's move on to part B. Here we go. For this one, because it's so long, I'm going to try to kind of draw out what I know as we go through the problem sentence by sentence, rather than draw the diagram or whatever, write down whatever I need to all at the end. So growth hormone stimulates liver and bone and other tissue to produce IGF. So we can write that down like this. So growth hormone stimulates liver and bone. And they go on to make IGF each. And the IGF from the liver cells is endocrine and stimulates growth of many cell types, including bone. So this IGF here is endocrine. I'm just going to write endo for endocrine. The IGF from the bone cells acts as a paracrine and stimulates bone growth. And this is paracrine. and it's only basically in the near local bone tissue. Both the paracrine and endocrine effects of IGF are required for maximum growth. And some very short children have low levels of IGF and don't respond to added growth hormone. Researchers have suggested treating the children with IGF instead of GH. It is possible that IGF will inhibit their growth, and why is that? So essentially, what the researchers are suggesting is we're going to artificially increase the IGF concentration in the blood, basically, because that's what injection does. And if you remember, that's exactly what endocrine means, hormones in the blood. So you would increase, right, this IGF here, but not especially the IGF. For example, the IGF that would originally have come from the liver instead of the IGF that comes from the bone, because the IGF that comes from the bone is only found in local bone tissue, and that concentration isn't affected by injected IGF. Then you can imagine this IGF that's increased specifically in the blood, the endocrine IGF, would cause a negative feedback loop, right, against growth hormone like that, and thus decrease the IGF that the liver itself is actually producing and then outputting into the blood. Okay, let's start going through each of the answer choices. Injecting IGF could inhibit growth by inhibition of blank. Now the key word here, I think, is not what is inhibited, but specifically it's which of these when inhibited would actually cause issues with growth, right? So the first one, protein synthesis in bone. Remember, injecting IGF doesn't affect the concentration of anything that has to do with bone. It specifically interacts only with the endocrine IGF and thus the liver that produces the endocrine IGF. It doesn't really affect anything with bone. This bone isn't really part of a negative feedback loop, therefore. So I would rule that out. Second is growth hormone synthesis. And that's exactly what this arrow, or this inhibition arrow, indicates. With high artificially high endocrine IGF, you would have decreased GH synthesis or release. So I would circle that, or I would choose that as one of the answers. Second, IGF synthesis in the liver. Now let's think about this. Sure, it's true that when growth hormone goes down, the liver's production of IGF should also go down. But does that actually affect growth negatively? And the answer is no, because we have this IGF that's already so artificially high, right, from the injection. So even if you have slightly decreased production from the liver, you still have plenty of IGF that's circulating in the blood. So I wouldn't choose that. Next is GHRH release, growth hormone releasing hormone. I don't necessarily see that in this diagram, right? This IGF specifically seems to be interacting mostly with growth hormone, and consequently maybe the liver. But I don't know about GHRH, so I wouldn't choose that for now. Somatostatin release. You may not know. First of all, somatostatin's not here. It's not anywhere in this diagram. But I think, and if you look back to the lectures or the notes, somatostatin, if I recall correctly, is something that inhibits growth hormone. And again, there is no indication that artificially high IGF, would lead to somatostatin release. So I would rule that out. Essentially, I would only have GH synthesis. Now let's see how we did. Again, same as before, we've got half of the answer, which is GH synthesis, but we also missed out on GHRH release. Now let's go back to our diagram and see why that's the case. Now remember, hormones, most of these final effector hormones like growth hormone and IGF usually are produced when their respective glands, for example, GH being produced from the anterior pituitary, when their respective glands themselves are activated by tropic hormones, right? And in the case of GHRH, that would precisely be in the case of GH, the tropic hormone would be GHRH, and which is released from the hypothalamus towards the anterior pituitary, right? And then you can imagine that there's another negative feedback loop that goes like this, because the IGF in the blood should also be picked up by the hypothalamus, and that makes perfect sense. Just like how the anterior pituitary can pick up the high IGF in the blood and stop GH production, the hypothalamus can pick up the high IGF in the blood and stop GHRH production or release. And so both of these combined their inhibitions would have the same effect, and that would definitely negatively inhibit growth. All right. So again, what do we want to take away from this? Now, on the left-hand side, again, we have the statement or the lesson that we shouldn't be taking away, which is because it's far too specific. Namely, injecting IGF into the blood via negative feedback reduces both GHRH and GH levels. Again, everything's correct. It encompasses both GHRH and GH, both answer choices. But in that first part where it says injecting IGF into the blood, like that is so specific to this problem. And it's hard to imagine that another exam problem or a later less learner's manual problem will have this exact experiment where they injected IGF, artificially high IGF, into the blood in children with GH production issues like that. This is hardly applicable to any other problems. But let's see what I would. Let's see what I would write down, or if I were the one trying to take away the lesson from this. Again, I have in green the concept, which is negative feedback in endocrine system potentially affects all upstream hormones, including tropic hormones, GH and GHRH, right? So even though GHRH is never explicitly mentioned, again, because its only effect is really to start GH production, and GH is really the one that's important in all this growth control. But because GHRH is a tropic hormone for GH, and because the hypothalamus, which produces the GHRH, can sense the high IGF, it also participates in negative feedback loops. And that's a common theme, right? That's a common general principle of the hormone unit of all these endocrine loops and circuits, is that there's multiple feedback loops. And you want to consider all of them. And the way to do this is by drawing this entire tree out, not just starting with the tree of hormones and like the endocrine pathway out, and not just starting with GH, but thinking about first, is there anything above or upstream of GH? And yes, there is a tropic hormone, GHRH, right? And by not drawing that or thinking of that, I missed out on half of the answer. So, and then the action takeaway in blue is to make sure to check for tropic hormones and draw out the entire endocrine cascade, not just what's mentioned in the problem, but with hormones, it turns out we want to map out all of the relevant circuitry, even though it's not explicitly stated in the problem. Okay, so let's move on to part B2. The question asks, if growth inhibition occurs, it will be as a result of interfering with which of the following, or which of the effects of IGF. Now, based on B1, the previous problem, and the diagram I just drew on the right here, it seems that most of the negative feedback, in fact, all the negative feedback loops seems to be happening through the endocrine pathway on the left of the diagram. The liver produce IGF, the GH and the GHRH, being inhibited. Right. Meanwhile, the bone produce IGF, and all the paracrine effects of IGF seems not to be too involved in this negative feedback. So I'm going to go with endocrine, since that seems to be the part of the hormonal circuit that's involved in negative feedback and all this modulation. Well, it turns out I chose the opposite answer. Now let's think about why that is the case. If you inject high IGF, right, and you do get low GH and low GHRH, right? Let's think about what the effects are for having decreased GHRH and decreased GH. You would have decreased IGF from both liver and bone, because GH is supposed to stimulate IGF synthesis in both liver and bone. Now, is it really a problem that you have low liver-produced IGF in the blood? And if you think about it, it's not a problem because you still already have so much IGF in the blood, in the endocrine. So you're not actually affecting growth negatively. Meanwhile, the paracrine effects of IGF can only come from the IGF that's produced from the bone and not from the injected IGF. And the moment you decrease the bone-produced IGF, there is no other alternative presence or source of IGF available in the paracrine tissues, in the paracrine pathway of involving this bone, the local bone tissue. So it turns out, although yes, liver synthesis of IGF does decrease, that has absolutely no effect or very minimal effect. While the bone-produced IGF, once that goes down, there is no other substitute. So that's the problem we're going to face here in terms of the growing child. So that's why the paracrine tree is more important in terms of this growth inhibition, overall growth inhibition. So again, let's see what lesson we can take away from this. On the left-hand side, which is far too specific, we have injecting IGF does not affect endocrine effects of IGF while inhibiting paracrine effects of IGF. Again, this whole injection of IGF is specific to this problem, and there's no guarantee that we will see this exact same setup with the exact same set of hormones in an exam problem. So that's far too specific. And to generalize here, I have three takeaways that I've written down. The first is negative feedback. Has to do with negative feedback. Non-tropic hormones in the blood inhibit the production of its upstream tropic hormones and itself. Right. So that's IGF. IGF is non-tropic. And for example, IGF is non-tropic, and it inhibits the upstream tropic hormones like GHRH and also GH, which is also non-upstream. And because of that, you technically get a decrease in IGF production, right? However, the second principle says if the non-tropic hormone is destined for the blood, then injection makes up for negative feedback, right? Any injection will increase the endocrine levels, concentration of the hormone. So, and that's why we have in this very problem no issues with having decreased liver produced IGF, because the injected IGF is counted as part of the endocrine hormone concentrations. And finally, if the non-tropic hormone is destined for elsewhere, then injection cannot make up for negative feedback, and that's a paracrine aspect, right? There is no other substitute. You can't substitute in the paracrine hormones with injected hormones. Because that's not how it works. Because the injected hormones will only reside in the blood and be counted towards the endocrine concentrations of the hormone. Notice that I didn't really mention anything about GH or GHRH or IGF in the three generalized takeaways, because these three principles can technically be applied to any similar setup of injecting hormones. That doesn't it could be, for example, something with like cortisol. It doesn't have to be with GHRH or GH or IGF. Okay, so let's move on to the second problem 907. This one has to do with muscles and their relationship with neurotransmitters. Each neuron releases one major neurotransmitter. However, many neurons release several co-transmitters along with the major transmitter. Some neurons that synapse on vascular smooth muscle cells release both Ne and ATP. Both ATP and Ne are found in the same synaptic vesicles. The ATP receptor in the muscle is a ligand-gated cation channel that is permeable to both Na+ and Ca2+. In a normal organism, part A, in a normal organism, both Ne and ATP are released together. Suppose we set up an experiment and add only one or the other to the muscle cells. So A1, which will produce a faster response. So I'm going to try and I'll see if I can draw this out. Let's go with each of these channels. So here we have the double leaflets of a plasma membrane. I'm going to draw two of them, one for Ne, one for ATP. So the ATP receptor is a channel. It's a directly linked channel, right? And has a binding site, again, for, let's say, ATP, which we'll draw in red. At which point cations will rush in. They specifically mention Na+ and Ca2+. So cations will rush in like that, okay, upon binding of this red molecule of ATP. Now let's draw the same for this norepinephrine molecule. Now is norepinephrine, are these, what kind of receptors are these, right? And unlike the ATP receptors, they're not actually the receptors themselves are not channels, essentially. So they're not ligand-gated cation channels, but instead they have usually a separate receptor, often a GPCR, which means there's an associated G protein, and then there's probably a second channel that lets in the actual cations. And the G protein helps activate or open the channel. And on here we have a binding site for, let's say in this case, blue for norepinephrine. So which will produce a faster response? Now, it seems to me really that we're looking at both, both involve a single molecule of ligand binding, and both involve one channel opening and cations rushing in. So I'm going to go ahead and predict both at the same time, although they seem to be going through different mechanisms. And it turns out that's not the case. Now, why would that be? And the answer is kind of already in the diagram that I drew. I drew the correct diagram, and I should have really picked up on this. But if you, the fact that there's simply more steps that the GPCR and the G protein have to go through before you can really open the channel itself means that it will take longer. Whereas in the case of ATP, all you need is ligand binding. There are no other proteins involved. The receptor itself is already the channel, so you don't need to activate a separate G protein. There doesn't need to be a secondary messenger cascade, which is often the case with G proteins and GPCRs. So already we're saving ourselves so much time here, and that's why ATP will end up producing the fast response. Now let's see what we can take away from this. So in terms of the lesson that we're learning, so ATP induces. So the two specific lesson would be ATP induces its effects faster than any receptors in smooth muscle cells. I meant to write ATP receptors. And again, the comparison between ATP and NE receptors is completely true and correct. But there is no, again, we can only use this if the problem specifically asks for, again, to compare the speeds of ATP receptors and NE receptors. And that's not always the case. We need to be able to compare the speeds of any any pairs of receptors that's presented in the exam. So in the green, we have the content lesson, which is ligand-gated ion channels, or so-called direct or ionotropic channels, are faster than indirect channels, or indirect or metabotropic channels. And the indirect example, the key example of indirect is GPCRs, because they have a G protein activation step and they have a secondary messenger cascade step before the cations can actually rush into the open channel. Again, so, and then the action lesson in blue would be to make sure to check the mechanism of the receptor, especially the mechanism of its action, to determine basically how quickly it can let in or let out certain ions, and basically how quickly it will respond upon ligand binding. Okay, so let's move on to problem A2. The problem A2 asks, if the muscle cell plasma membrane contains voltage-gated Ca2 plus channels, they can probably be opened by addition of. Well, I'm thinking, so voltage-gated Ca2 plus channels, or in general, voltage-gated channels open when there's a change across the voltage of the membrane they're part of, right? It just so happens that in this case, Ca2 plus ions will be moving in when this voltage-gated channel opens. Well, looking at what the effects of both the NE receptor and the ATP receptor, but in both cases, cations are going to rush in, or there's going to be some form of membrane change or membrane voltage change. So I'm thinking that either one can actually can cause the necessary voltage change to open up this voltage-gated Ca2 plus channel on the plasma membrane. It turns out, that's not the case, and let's think about why. So the ATP receptor is a simple ligand-gated cation channel, which means ligand's going to bind on the extracellular side. The channel itself, which is on the plasma membrane, will open up, and the cations will be moving across the plasma membrane. Now that's a key point. Because the cations are moving across the plasma membrane and not any other membrane, that's why we're going to observe a voltage difference across the plasma membrane, and that's what opens up voltage-gated ion channels on the plasma membrane. It's the specific voltage difference between the inside and the outside of the intracellular and the extracellular side of the plasma membrane that opens up these voltage-gated Ca2 plus channels. Now, why is not the case? Then why don't we see that with the GPCRs binding NE? Well, it turns out, unlike the way I drew it, the cation movement is actually through the ER, meaning when the NE molecule binds the GPCR and the G protein is activated, it's going to go through this IP3 pathway. Essentially, and the endpoint of the IP3 pathway, it turns out, is the fact that is that Ca2 plus channels that's stored in the ER, it's going to go into the cytoplasm. So you're really seeing ion movement across the ER membrane, but no ion movement across the plasma membrane, which means the voltage-gated channels, which are specifically stated to be in the plasma membrane, will not be open because there is no movement of ions across the plasma membrane, and thus no voltage difference occurring. And that's why only the ATP receptor, which is the only receptor that lets in ions through the plasma membrane, is the one that we want to open the voltage-gated channels. Now again, the far too specific lesson in this case would be ATP alone is enough to open voltage-gated Ca2 plus channels on smooth muscle cells, while norepinephrine is unnecessary since it uses Ca2 plus from within the cell. Okay, that does so. Not only is that not too specific to this problem, but it doesn't explain really the rationale, the general concept behind why the ATP alone works while the norepinephrine receptor doesn't work. That's better explained in the two generalized principles on the right. First of all, voltage-gated channels often come in tandem with ligand-gated ones, right? And so that generally established the fact that, for this voltage-gated channel to open, you need some form of membrane voltage difference, and that has to happen initially. That has to be set up initially by a ligand-gated receptor of some sort that opens a channel across the plasma membrane specifically. Now, the second principle, which is more relevant to this specific question, is internal ion movement rarely affects the plasma membrane voltage difference. Internal ion movement, such as movement across the ER, right, instead of the PM. And that, of course, the ion movement doesn't affect the plasma membrane voltage difference. And so only channels that move ions across the PM are needed specifically to open voltage-gated plasma membrane embedded channels. And that's why ATP receptor does that. So that's the only one you need to open these voltage-gated channels, and not norepinephrine receptor, because that only causes internal ion movement within the cell. Okay, let's move on to problem B1. So B1 is a pretty short one. It asks ATP alone will probably cause blank. And in the previous problem in A1 and A2, we've established that when ATP binds to its surface receptor, what happens is that Ca2 plus will enter from the extracellular side of the plasma membrane into the cytoplasm, essentially, and this will of course cause a depolarization and also the opening of more voltage-gated channels, letting in more cations into the cytoplasm. Which means we know from our muscle unit that if you have an influx of Ca2 plus into the cell, the Ca2 plus is used to contract the muscle. In this case, smooth muscle will probably, which means the smooth muscle around the blood vessels will probably end up constricting or cause vasoconstriction. Let's see if that's correct. This one turned out to be correct. It was a fairly short one and pretty simple one, and I wanted to show you what you should be thinking of if you get the question right. Now, there's some instances where you might get the question right, but because of the wrong thought process, or because you were essentially lucky, or relied on some other crutch that isn't really the right way to think, or the way the solution manual wants you to think. And in that case, then you should treat the question as if you got it wrong, and again go through your lesson log and jot down a new entry. In this case, it turns out we had the right thought process with the influx of Ca2 plus causing muscle contraction and thus vasoconstriction. So I won't be writing anything down, but I just wanted to show you what would happen and what you need to be weary of even when you're getting things right. Okay, let's move on to the last problem, B2. In a normal organism, when these neurons cause the vascular smooth muscles to contract, the Ca2 plus needed for contraction should come from blank. Well, we've been spending this whole problem talking about how ATP release from as a neurotransmitter and ATP binding onto the receptors of the smooth muscle cell causes depolarization across the plasma membrane, and thus opening of more voltage-gated Ca2 plus channels on the plasma membrane to let in Ca2 plus from the extracellular side into the cytoplasm. And then all of that causes vasoconstriction. So I'm thinking the answer must be outside the cell. Well, let's see if we're correct. Well, the answer is both. And now I'm really confused because, again, as I said before, we spent most of the problem beforehand talking about how the ATP is the one that really matters, and ATP specifically brings in Ca2 plus from outside the cell. So what's going on here? Why is it not just outside the cell? Why is it both? Well, this has to do with the question stem. And to look at the first four words of B2, it says in a normal organism, right? They're talking about real life, not like in an experiment. And right above that, I have also the question stem for A, part A, and look at the first few words again. In a normal organism, both NE and ATP are released together. So let's draw this out like schematically. So in a normal organism, so something like normal, in a normal organism, it's both Ne and ATP, right? Combined. So something like that. It's both. And yet, so what was this discussion that we had about specifically ATP being released? Well, look at the second sentence of A. Suppose you set up an experiment and add only one or the other to the muscle cells, right? So in the experimental condition, in the EXP condition, we were only using Ne or ATP. Never both, not both. Right. So this tells us something. So all the problems that we had about, is it Ne that's important or is ATP that's important? Well, that we were only asking these questions in part A because part A specifically tells you this is not in a normal organism or in real life, but rather in an experimental condition where you're only adding one or the other. But of course, part B is talking about in real life, in which case both are always involved, which means you can envision, ATP causing Ca2 plus from the outside, bringing Ca2 from the outside, so that would be extracellular Ca2 plus. Well, Ne would bring in Ca2 plus from the inside, from the ER, right? And that's why you would have both extracellular and ER, in which case the answer would be both. Okay, so what do we want to write down here? So the much too specific answer, or the lesson to take away, would be in a normal organism smooth muscle cell, Ca2 plus from both outside and the ER causes its contraction. Again, that's and this is true. This explains the answer, but it doesn't really tell you, how we were supposed to get to the answer, right? So it's not really an applicable principle that we can use for other problems. So let's look at what I wrote down, or what I would write down if I were the student solving this problem for the first time. So the first one's very similar to like kind of what we had on the left hand side. Smooth muscle, unlike skeletal muscle, uses Ca2 plus from both ER and extracellular space. Okay, so that's really not much different. You're asking why did you put this here? Well, to begin with, this is the kind of like the factual stuff that I want to make sure that I got. And again, this isn't going to be the main takeaway. The main takeaway is going to come later. But just having this fact down, I think would be I'm suspecting might be helpful in the future. Second, in a normal organism, both ATP and Ne are released at once onto smooth muscle cell. So this is kind of like the other part of the problem that we looked over because we were so focused on Ne versus ATP from part A. And really, it's the fact that both ATP and Ne are released at once onto the smooth muscle cell, right? And even then, you might ask, well, June, like this, they might not even ask me about ATP and Ne combination, right? Then how is this? How am I supposed to take away a really generalizable lesson? And I think that's the last point here, and this is probably why and this is the main crux or the reason why I kind of missed. I couldn't get to the answer in the first place, which is make sure to distinguish experimental conditions to the normal situation or the real life situation seen in nature, right? And that's exactly why I missed it. We were confused because we thought the experimental condition of applying just one or the other neurotransmitter could be applied from part A and to part B. But really in part B, they were asking what's going to happen in real life. And notice that's a lot different from what you see in experiments or the laboratory. So the blue part is again, the blue statement is the action plan, what I need to be thinking about and how I want to approach the problem next time. And this really is applicable to anything. Always make sure to understand what conditions you're working in, whether it's experimental or if it's natural or if it's a new condition or if you're going back to an old condition that you used that was introduced to the problem in an earlier part, right? So make sure to keep that in mind. So that concludes the contents of the video. I hope that you found it helpful. I know we've only gone through 2 problems really, but each of the problems had multiple parts. So I hope that you were able to see many of the ways, or at least some of the ways that you could apply this kind of lesson log, generalized lesson log strategy to the learner's manual questions. And again, to summarize the main takeaways from this video, is first of all to think in terms of large concepts, think big, and not the minute details of each problem. And that's because you will never see the same problem from the learner's manual or an old exam in your actual exam, or elsewhere. You know, each problem is so unique, but these problems are not completely random. And they're not completely different from each other. They have general threads, common threads of ideas that you can apply to each of them, and that's kind of what you need to dig out, right? And especially the ones that you kind of miss or you can't think of or you're confused about, those are the ones you want to write down in your lesson log. And another way to phrase that is, you know, always ask yourself when you're trying to write down in your lesson log, can I apply this? Can I apply whatever I'm writing down to a slightly different problem? Right? And with the examples that I gave with the much too specific lessons or, you know, takeaways, you really can't, because those sentences, those lines include details that are so specific to that problem, that they're not too useful outside of this specific problem's context. But always asking this question and trying to take away the most general lesson and the larger concepts will help you in terms of figuring out a way to tackle new problems without making the same mistake. And again, I wanted to thank you a lot. This is the first time I've done this. I've never made a video before of this sort, and this is also the first time the bio department, especially the intro bio class, has done this, where we're using a video to kind of convey how you would like students to approach the problems, not in terms of how to get it right, but more importantly, how to learn from them. Because again, that's the hardest thing to do. It's once you know how to get things right, it's pretty smooth sailing from there. But the challenge is always to figure out how do we want to learn from the problems we miss. So yeah, this is the first time any of the instructors or the TAs have done for this course. And I would love to hear your feedback on how I can improve. Ideally, I would like to make another video this time for the fall semester learner's manual, so that the incoming students can benefit from this immediately, before they even take their first exam. So yes, please send your feedback, your thoughts, constructive criticism to this email right here, yk2611@columbia.edu. That's my email. And I'm June, BioTA. You can also contact me directly if you're in my section, by coming to my section. All right then. Well, thank you very much, and good luck with your studies.